I'm very honored to be giving my presentation to you. And I'm Atsumi from Synthetic Gestalt, an AI company. And my presentation will focus on the uh, commercializing or utilizing minimal cells to produce various use of products. And uh, to realize that we edit the metabolism pathway and with generative development and medium optimization. Ultimate goal is produce various chemical products and like food ingredients, fuels, industrial chemicals, or industrial materials, and and more. Uh, for example, more concretely, we are focusing on four products: Kishitol, Marit, Barin, Panthenate. They have a practical use, and they will contribute the self-efficiency of minimal cells. For example, Kishitol and Marit is usually revealed in food ingredients, and barin is also found in food, and barin is more profitable than kishitol and mallate because barin is contained in a supplement known uh, as a BCAA, and also pantothenate is known as a vitamin C. Sorry, vitamin B5, and varying and pantothenate will contribute the self-sufficiency of the minimal cells. But how to realize these products? To realize these metabolites, we edit genetic conditions and optimize medium conditions. But they are a bit challenging. So, in this context, using minimal cells is very important, we believe, because once the once we establish the process of metabolic production, that minimal cell will be the best platform to know what protocol is best, what protocol is required for getting desirable metabolites. Because minimal cells, of course, have minimal components, so we can reveal what is going on. So I believe that the minimal cell will be best platform to find the protocol to realize several production. And we have two aspects, gene optimization and medium optimization. And gene optimization is normally done by scientists, though digital system can assist them. On the other hand, if we focus on media optimization, that a digital system can be the best practice to find the optimal condition. Because the digital system can simulate the uh, various conditions of media. And what is a digital system? The entire experiment system consists of wet cycle and dry cycle. And we have a dry cycle. Since the Gestalt is contributing dry part of entire system, uh, and we we have developed a platform of that digital twin with Dr. Yo Suzuki, and we are focusing on the simulator that is located in part of dry cycle. So how to realize such a simulator? We have three processes. Pathway simulator construction that will make a backbone of simulator, and next we will refine that. We refine the model with experiment data that is enhanced by machine learning technique. The last one is execution of simulation. We can execute the simulation and compare various results. Firstly, we have done pathway construction. We can do that automatically because we have established a digital system which can produce the pathway backbone automatically using a list of enzyme and public database. Once we can decide the range of used enzyme, we can automatically produce the backbone of simulator. But this is the most important part. Even if can produce the backbone of simulator, it is not always perform precise simulation along with experimental data. So we have to tune up the simulator with experimental data. So we employ the machine learning technique, data assimilation, using experimental data, and then the simulator result will fit the actual experimental result. So this is one of the simulator tune-up, and configuration is updated. So once we get the tuned up model, refined model, we can move on to the dry experiment. So we prepare the various conditions having different media condition, and then we can do the simulation simultaneously, parallelly, the hundreds of models, and then the hundreds models will return the result. That means that final product metabolite, final state of metabolite, and we can easily find the best result of metabolite among these models, and we can reach the ideal conditions producing aiming ingredient. That of course that assists the experimenter who and the experimenter can find what condition is best to grow and to produce aiming production. This is the entire our system, and we are aiming the useful ingredients. And I explained about construction, refinement, and the execution of simulation that helps the experiment. This is because sorry. This is why the digital system we have assist contribute the entire experiment process to produce aiming compound. That's it. Thank you for listening.
Can you comment on any progress that you've made in your collaboration with Yo Suzuki at JCVI?
Yep, of course. Thank you for asking. And we developed the pathway simulation and refining system. So once Dr. Suzuki can get the result of gas chromatography, we can fine-tune we can tune our model. So we Synthetic Gestalt and Dr. Suzuki have tried to get the first result of GCMS, gas chromatography, mass spectrometry. So we have ready to improve our simulator, and it's maybe does that make sense about
Yes.
your
Yes.
question? Thank you.
Thank you very much.